Okay, lecture 6, we'll talk about mutation and DNA repair mechanisms of different kinds. In biology, mutation is very important because they are the foundation of the theory of evolution, as you know. The DNA, what you have in your cell, are very important, especially the integrity of it. Okay, so they have to be maintained from generation to generation. That's how the hereditary things are passed down to generations. But it's not always maintained the same. There are some changes made, as you know. But how are the changes made? So these are different ways of making changes. Cells go through cell division. Before that, DNA molecules replicated, as we learned from the previous chapter. But that DNA replication process is not perfect. Okay, DNA polymerase enzyme is not perfect, so there are some mistakes made. Okay, so any inaccuracy in the process of DNA replication will cause the change of the DNA, so that's mutation. And then exposure to some chemicals, okay, so that can change the structure of your DNA, especially the base parts. So-called mutagens are the chemicals that can damage the integrity of your DNA. Then, yes, if that DNA is used as a template for DNA replication, of course, there will be changes made. And then insertion, or maybe deletion of DNA elements could be done. Okay, so then of course that will cause that mutation. Okay, so there are many types of mutation possibility basically. Then if there are so many possibility of mutation, then how would the cells be able to survive? That's kind of question, right? But the thing is, cells have some means of recognizing errors or changes, and then they have a mechanism to repair those changes made on their DNA before they are passed down through the DNA replication. Okay, so those are DNA repair mechanisms. Okay, so understand that. So that's what we're going to talk about through this lecture. So, and then when you say, oh, DNA is changed, then what do you mean? I mean, if you think about the structure of the DNA, DNA is made out of nucleic acid, and nucleic acids are made out of nucleotides. If you look at the nucleotides, the identity of the DNA is normally defined by what bases they have, right? Because phosphate sugar backbones are pretty much the same regardless of what nucleotide you have. The only difference comes from the bases and their, the sequence order that determines the identity of your DNA molecule. So the changes are mostly happening around the base structures. Okay, understand that. So the definition of these words, mutation, okay, means any heritable change in the base sequence of DNA. That's what mutation is. Makes sense. And the mutagens may be physical or chemical agents that can cause mutation, basically change in your bases. Okay, and the mutagenesis is the process of establishing or producing a mutation. Okay, it can happen naturally, or it can happen artificially through induction, meaning exposure of your cells to the mutagenic conditions, like strong UV light or with the presence of some mutagenic chemicals. So spontaneous mutation means that that's naturally happening mutation. Okay, and then induced mutation means it's artificially caused mutation by exposing maybe cells to the mutagenic condition. Okay, and then a point mutation means a single base pair change in your DNA. Okay, if you change a single base pair, maybe a single nucleotide, what are the consequences of that? Maybe your base can be changed through a substitution. Maybe like this case, AT was the original base pair that was in your original DNA, but let's say A was changed to G, then its pair will be also changed to C. So that's the substitution, right? Or a point mutation could be made through addition of a single base pair or deletion of them, okay, which will also cause a drastic change in your DNA. Okay, so let's think about maybe changing, switching out one base pair, okay, to another. Okay, then what are the consequences of that? If you change, let's say, one nucleotide to something else, maybe if that nucleotide happens to be a purine, like adenine or guanine, okay, if that adenine is changed to maybe another purine, maybe A is changed to G, okay, or pyrimidine to pyrimidine, okay, is maybe T or C, okay, between the change of these two, we consider those changes as transitional mutation. Transitional meaning, oh yeah, they are changed, but within the same group of nucleotide. Okay, so the result may be minor compared to the other case. Okay, which is transversional mutation. That's a switch between two different base groups, like purine to pyrimidine change. Okay, or pyrimidine from pyrimidine to purine change. Okay, so understand the definition of this: transitional mutation versus transversional mutation. Okay, and the result of mutation in the protein. So if your gene happens to code a protein as its final end product, okay, so the change on the DNA will affect what protein will be made, right? Especially in terms of the amino acid sequence. Let's take a look at that. Okay, so the I guess the minimum consequence would be the silent mutation. Okay, so yes, changes made in your DNA, but the change was not made at the DNA, the protein level. Okay, because the change within the DNA sequence was minor. So that was the original, let's say, DNA sequence, which is transcribed into this sequence, you know, T to A, A to U, because there's no T in RNA, right? And then C to G, and A is to U's, and C to G. So if you have that messenger RNA sequence, if you look up the codon table, you will see AUG is the methionine, the start codon, and the UUG is the phenylalanine, and GUG is glutamine, and then UAA is tryptophan, right? So you'll see that. And now you say the DNA sequence is changed, so C, the original nucleotide, was changed to G there. That was the mutation. Okay, and then if that is transcribed into an RNA, you'll see, you know, everything else will be the same, but G, which was changed from C, will be transcribed into C there, right? And the question is, what is that codon UUC? Okay, what amino acid will be translated from that codon? If you look at the codon table, UUG and UUC, they both translate the same amino acid, which is phenylalanine again. So if you look at the sequence of the protein, amino acid sequence, it'll give you the same amino acid sequence. So at the end, the product, there's no change. Okay, methionine, phenylalanine, glutamine, and tryptophan. There's no change on the amino acid sequence. If that is the result of mutation, we call that a silent mutation. Silent mutation. So it's silent because there is no mutation effect at the protein level. Okay, understand the definition of that. And the next one, we call that neutral mutation. Neutral mutation. So look at the diagram again. You know, so original sequence and the RNA protein sequence. Now you have a change on the first nucleotide of the codon. AAC was changed to GAC. You understand, you know, if you look at the codon sequences, you know, the third nucleotide can be variable, but still they may code for the same amino acid. But if there was a change made on the first and second, it's most likely the amino acid sequence will be changed. So in this case, you know, A to G change made a change on the protein sequence, because UUG, the codon on your mRNA, changed to, you know, CUG. Okay, so UUG again was the phenylalanine, but CUG codes for leucine. Okay, so it's from phenylalanine to leucine switch is what you made. Okay, so if you remember the different groups of amino acid, You'll understand phenylalanine belongs to the group of hydrophobic amino acid, right? How about leucine? Yes, leucine is also a hydrophobic amino acid. So it's a change between hydrophobic amino acid to hydrophobic amino acid. So, you know, the chemical property-wise, there isn't much change. So hydrophobic amino acids tend to be buried when this particular protein is folded. So it's not going to cause a drastic change in terms of how protein will be folded and how its structure will be, you know, maintained or changed. So there isn't much structural change that's expected. So if that's the case, then we will consider it as a neutral mutation. Neutral. Why is it neutral? Because, you know, amino acid, the characteristic is not changed much from the same characteristic amino acid. The switch was made within the same property of the amino acid, hydrophobic to hydrophobic change. Okay, so that's considered a neutral mutation. And then how about missense mutation? You will expect to see a little more drastic change. Yes, that is the case. So again, the original sequence, in this case, let's say there was GTC was a DNA sequence which is transcribed into CAG, okay, which codes for glutamine. Now GTC is changed to TTC. Okay, TTC will be transcribed to AAG, and which codes for lysine. Okay, so it's a switch between glutamine and lysine. Okay, so then what kind of amino acid is glutamine? Glutamine is probably you know, hydrophilic polar amino acid. And how about lysine? Lysine is one of those three positively charged amino acid. Okay, from hydrophilic amino acid to positively charged amino acid. So that is a more drastic change. So there will be a change on its structure, and the protein may have different structure and function through this mutation. So if that's the case, we consider that as a missense mutation. Missense mutation. Okay. And then finally, there is something called nonsense mutation. Nonsense mutation may be another drastic change. What it's going to do is, of course, the codon switch from GTC to, the DNA sequence change from GTC to ATC, so from G to A change. And if you look at the codon sequence on your mRNA, it's CAG versus UAG. So CAG glutamine, but UAG is what? It's one of the three stop codons. So stop codon will bring in what? There's no amino acid that's going to bring in. So instead, your translation will be terminated as soon as there's a stop codon there, because there's no tRNA they can recognize and bind on that stop codon. So your translation is early terminated, then what? This protein will never be made in your cell. So actually that's the eliminating, how you can eliminate that, you know, gene product. So we call that a nonsense mutation, which causes, you know, elimination of protein synthesis because there's a stop codon added. Okay, so very drastic mutation. And then finally, there's another mutation that causes drastic change, that is called frame shift mutation. Frame shift, you know, that is made through either addition or deletion of a single nucleotide, okay, on your DNA sequence. So let's see that, you know, that is a DNA sequence translating to that mRNA sequence, and it's going to give you that protein amino acid sequence, right? But now the change is made from T to G here on that codon on your DNA sequence, and that's going to give you that mRNA sequence. Okay, I'm sorry, that's not the change. I guess there was addition of G between G and T there. Okay, there's an extra G nucleotide added there. Okay, so when you add that, so that is transcribed into that mRNA sequence. Then think about it. The codon, it's a triple nucleotide reading frame, right? Every triple nucleotide, you have amino acid brought in. But, you know, after that single insertion of a nucleotide, what happens? So the sequence of codon is switched drastically because it's shifted around. So we call that a frame shift, because the insertion, you know, the deletion would cause the same thing. So instead of, you know, you have CAG, you have CCA codon. After that, you have GUG codon and then GUA codon. So everything is changed after that insertion. Okay. So if you look at the protein, yes, I mean, the sequence will get changed continuously. So that is considered as a frame shift mutation. Okay.